Okay, so first of all, let us look at these phloem tissues. So what is their functions? So generally in terms of phloem tissues, their function is to conduct the dissolved organic assimilate. Okay, dissolved organic assimilate. So from where to where? From source to sink. Okay, so in terms of tissues, you can see that if phloem tissues is not only consist of one type of cells, but all kinds of cells. So we do have first sieve element. So if you put side by side, it's comparable to the xylem vessel elements. Okay? Next, we have companion cells. Special only happens in only occur or only can be found in the phloem tissues and phloem fiber, phloem parenchyma. So as we have gone through the xylem, if you compare to the xylem, so xylem also have four kinds of tissues, four kinds of cells, sorry. So we have one, we have the xylem vessel element. Okay, number two, we also have the trachea. Number three, okay, they also have the xylem fiber. And number four, xylem also have the xylem parenchyma. So in terms of the role, actually fiber for supports, they are the same. You compare both of them for support. Can you see that? For xylem parenchyma is for storage. So it means that this one also for storage. So no different among them. Okay. And tracheids, we don't learn them in our A-level syllabus, but xylem vessel elements is the transport, involved in the transport, and sieve elements also involved in transport. It's just a difference. Transport of what? Xylem vessel element transports the ions, water. Okay, sieve elements transport dissolve. Okay, highlight the word here, dissolve organic assimilate. Okay, most of them, they are photosynthetic product. So in our syllabus, we are going to use sucrose as our model to study how the transportations of these dissolved organic assimilate. So when say dissolve here means that water soluble. Okay, so we're going to look at why water soluble. If not water soluble, we wouldn't be able to transport them. Okay, so how we identify the cells? Let us look at this before we identify them the cells. So let us look at sieve element. Sieve elements initially they are the elongated cells stacking on each other. So let us look at three cells. Okay, 1, 2, 3 cells. So they are stacking on each other. Okay, so they have the cell wall. They have the cell wall. They have cell wall. Now, compared to the xylem vessel elements, okay, you can see that later I'm going to compare. Okay, so for sieve elements, they will have this what we call the perforated. Okay, perforated. Okay, instead of totally broken down, they are perforated. So if this is the another cells, you can see that they are perforated. So it means that they have a lot of this pore. Can you see that? Okay, if you compare. Okay, draw another one. Part A of it. So the end wall here, if you compare, I will write both. So this one is called as the sieve plate. So how you find it? You will see this sieve plate. So sieve plate actually is the end wall perforated with pore. Quite a large pore. Are you clear? They are quite large pore. Okay. So this sieve plate actually allows the assimilates to be transported. Can you see that across? Okay. So this. Okay. So the pore. So this pore is named as sieve pore. So sieve pore allowed assimilate to be transported along the sieve tube element. Okay, sieve tube elements. So in this case, if you compare, okay, if you compare to the end wall of the xylem, when you compare, question. So if you compare the end wall of the xylem vessel element, so the end wall of the xylem vessel element is totally broken down. Okay, so means that sieve tubes, if you look at this sieve tube element, they form column, but not a continuous column. Okay, but because of the end wall xylem vessel element totally broken down, so they form a continuous column. Okay, you look at the end wall, compare. Here, that's one. Second, you look at the wall. Okay, so the cell wall of the sieve tube element is unlignified. Okay, unlignified. But if you compare to the xylem vessel element, lignified. Okay, so if you look at this, again, we do know that if the presence of the sieve plate, the presence of sieve plates definitely, okay, I put a star shape here. I mean, with the sieve pore, allow the transportation, but they also, with the sieve pore, it provides, okay, low resistance. But if you look at the process, the presence of, okay, the presence of the sieve plate actually lower down the rate of transportation. Are you clear? If you look at the sieve pore, yeah, sieve pore do provide this low resistance for the transportation, but the presence of this sieve plate definitely will lower down the rate of transportation if you compare to the end wall that form the continuous column. So means that increase the rate of mass flow. You can see that. So the end wall xylem vessel elements here increase because it's totally broken down. But the sieve plate here, you can see that they were going to, even though with the pore, they reduce the resistance, but the rate still lower. So what is the function of sieve plate? So highlight here. Sieve plate actually provides support also. Why? Because no lignification. Are you clear? Okay, so I repeat again. So sieve plate is the end wall that perforated with a pore, and this pore is termed as the sieve pore. Okay, so this sieve pore allow assimilate it to be transported along the sieve tube element. It's kind of got the pore there. Okay, so if sieve tube elements, in this case, they only form a column, but not a continuous column if you compare to the xylem vessel element. If you compare here, the sieve pore definitely with the perforated, it provides low resistance, but no matter how the presence of the sieve plate reduce the rate of transportation, and this transportation by the mean of mass flow also. Are you clear? Okay, so if you look at this, if you compare the transportation rate in the vessel element, will be high compared to the sieve tube element, but we cannot get rid of the sieve plate because sieve plate provides the support, as we do know that the wall of the sieve tube elements, they are unlignified compared to the xylem vessel element, which is lignified. Okay, so now if you zoom into the cells, so realize that they are living cells with thin cytoplasm, peripheral, meaning at the side. Okay, I just draw for one cell, okay? So thin peripheral or thin layer of cytoplasm. Okay, if you compare to the xylem vessel element, you compare xylem vessel element, is a hollow tube with no cellular content. But for the for sieve tube elements, they have thin layer of the cytoplasm, okay, but okay, they don't have nucleus, they don't have ribosome. Are you clear? So this thin layer of cytoplasm is also to reduce the resistance or mass flow. Okay, you compare these three structure. Clear or not? Okay, so you can take a photo. Compare between the sieve tube elements and also the xylem vessel element. So if you look at the structure of the phloem, okay, phloem tissues, so now you can see that with the sieve plate and the perforated, so we know that these two must be the phloem sieve tube elements. Okay, so now we have another two cells here. They are living cells with a nucleus. Okay, one is a little bit more darker in stain. So because of the accumulation assimilate, so these cells called companion cells. I will discuss this in a later, right, at a later part. Okay, and then you can see that's a bit greenish in color, but actually it's not very greenish. Okay, in the real life, so you can see that in actual case, these living cells, okay, they are a parenchyma cells. Sorry, they are parenchyma cells. Okay, xylem fiber is not shown in this. As a xylem fiber, phloem fiber is not shown in this diagram. Okay, so we started with the sieve tube element, as I said. That's so their functions actually involved with the translocations. So initially, there are many elongated sieve tube elements joined end to end vertically to form a column, but not a continuous column. So they are living cells, as I say that, because no lignification, so they are living cells. So they do have the cellulose cell wall, they have the cell surface membrane, they do have the endoplasmic reticulum and mitochondria. Okay, but they form only thin layer, okay, with no nucleus, no ribosome. This is very important because nucleus is quite large, right? So to reduce the resistance for mass flow. Okay, so because we want to transport, right? So means that we want to increase the rate. So therefore, the end wall of two sieve tube element meets the sieve plate is formed. Okay, so this wall actually perforated by large pore known as the sieve pore. And then right here, so known as pore. Okay, so they are open pore. for the flow of the liquid through them. So you can see that, guys, in this diagram, you can see that the pore. So this pore allowed the assimilates to pass through. Okay, so if you have this is longitudinal sectioning, if you have the transverse sectioning, then you can see that the sieve plate with the dot actually they are the sieve pore. Okay, so again you can see that this is a transverse sectioning with the sieve plate, sieve plate. Okay, so sieve plate, the function of sieve plate also very important right now. Sieve plate also provides the support for the sieve tube element. Why? To prevent collapse. Are you clear? Because the under tension, like xylem is lignified, so not much issues. But for the sieve tube elements, it will collapse. It's something like pillar there. Okay, to make sure that when this sieve tube under tensions, it won't collapse. Okay. So if you look at this diagram, you can see that here with the structure of sieve plate, you can see that the pore again. So the parts, can you see that the part without the sieve plate, it collapses? Can you see that? Some part when under tension, sieve plate, the part that without sieve plate, it collapses. But it's not totally collapsed thanks to the presence of the sieve plate. Okay. Now, one very, very important cells at the phloem tissues is called companion cells. Okay, so these companion cells, you have to know that each sieve tube element must have at least one companion cell. Are you clear? At least one companion cell. So companion cells have a structure of normal living plant cell. So they have cellular cell wall, cell surface membrane, cytoplasm, small vacuole. They have nucleus. Are you clear? Okay. But one thing that you have to understand, both companion cell and sieve tube element, they don't have chloroplast. Right now, because their role is not involved in the photosynthesis. Okay, both companion cells and sieve tube element, they don't have chloroplast because they do not need to involve in the photosynthesis. Okay, now one thing special about the companion cells compared to the sieve tube elements, they have very high number of mitochondria and ribosome. Are you clear? So higher than the normal living plant cells because they are metabolically very active. Okay. So companion cells are very closely associated with their neighboring sieve tube elements with numerous plasmodesmata that actually join both cells together. So they allow a direct contact between the cytoplasm and companion cells. So what's the companion cells? Companion cells actually transport the product of photosynthesis from the source to the sieve tube element. Okay, they also involve the synthesis of the various proteins used in the phloem sieve tube elements. I will explain it later. Okay, so you can see that. Okay, this is the sieve tube element with very thin cell wall, and you can see that the slightly darker stain here. This one, they are companion cells. So each sieve tube element must have one companion cells. So same goes to here. You can these sieve tube elements. You can write down, guys. The histology, you can see the sieve tube element. Why we have a sieve plate? Can you see that? This is a sieve plate. Sorry, I used different color. So beside this sieve tube element, you can see that with the nucleus, right? So they are the nucleus. So they're living cells. So this one, they are the sieve tubes. I'm sorry, they are the companion cells. They are component cells. Okay, so now let us compare between the Sieve tube elements and also the component cells now. Okay, let's look at this Sieve tube element and also component cell. So I draw the transverse sectionings, transverse sectioning. So you have a Sieve tube element. So they have very thin cell wall. Because no magnification. Okay, so each Sieve tube elements they do have, each Sieve tube elements they do have one component cell. So the shape of component cells is quite small if you compare the Sieve tube elements, and the shape sometimes will be rectangular or triangular. Okay. They also have very thin cell wall. Okay, unlignified. Okay, unlignified. And then you can see that between, okay, write down, this is a Sieve tube element. Okay, this one is the component cell. So between the component cells and the Sieve tube elements, always you can see that they are having this plasmodesmata. Okay, so I label this one as a plasmodesma. Singular, okay, plasmodesma. So if you compare component cells, you can see that sometimes they have the nucleus. Okay, but Sieve tube elements no nucleus. Okay, so first we compare in terms of nucleus. So component cell. Yes, Sieve tube element, no nucleus. Okay, number two, we compare ribosome. Okay, component cell, yes, Sieve tube element, no. Number three, we compare mitochondria. Okay, component cells will have more compared to the Sieve tube elements. Are you clear? Both of them has. Okay, component cells have mitochondria. Sieve tube elements also have mitochondria, but in terms of number, the component cell will have more. Okay, then number four, if you compare in terms of the cytoplasm, Sieve tube element actually form a thin layer of cytoplasm, but component cells is normal. Okay, so means that you can see that if I draw out the cytoplasm, it will be somewhere here. Okay, very thin layer only. To luxury the cells. So let me have draw it slowly. Later you can take a photo, right? No worry about it. Can you see that? Okay, very thin. So what are the role of component cells? So this is the nucleus of the component cells. Okay, so what are the role of component cells? So basically, component cells involved in the active loading of assimilate. Okay, so role of component cells. First, loading of assimilate into Sieve tube elements, and number two, synthesize protein for Sieve tube element. Now, why? Why they need to do this? Because Sieve tube elements, they don't have the ribosome. They don't have the nucleus. So means that they don't have the gene to actually carry out the transcriptions and translation in the ribosome. So no nucleus, no ribosome in the safety elements, then no choice. They have to depend on the companion cell to synthesize the protein for the safety element. So in this case, it can be protein, it can be assimilate or photosynthetic products. Okay, so we can take a photo of these safety elements compared to the companion cells.